So thanks everybody for joining us today. We are going to be giving a presentation on comprehensive container qualification programs for deep and ultra cold storage products. So as we go through this presentation, we hope to provide some insight on the overall container qualification process and then focus really in on some unique considerations for deep and ultra cold storage products, especially as it pertains to container closure integrity testing. So here's a quick agenda for today's webinar. I will leave this up on the screen for just a moment so that you can take a look at it rather than me reading it out to you. At the end of the presentation, we'll also have some time for Q&A. So at that point, at the end of the presentation, just feel free to send a note in the meeting chat or to raise your hand via the meeting chat, and then feel free to turn on your microphone and ask questions at the end as well. Just a quick introduction to CS Analytical for those who aren't familiar. CS Analytical is a CGMP, FDA-registered, and DEA licensed laboratory that is exclusively dedicated and focused on container and package-related testing for regulated markets. So that's pharmaceutical, biotech, medical device, etc. The company was founded by folks with decades of experience in the contract laboratory space, specifically focusing on container and package system testing, myself being the shortest with over 12 years of experience specifically in this space. And so for CS Analytical, really our goal is to be able to provide end-to-end solutions for container package system qualification, starting with package development and supporting that product and that client through clinicals, through commercials, and up to expiry and use of that product package system. Really, one of our key differentiators is that we like to operate in a solutions-oriented collaborative approach. So really working with our clients, not only just to say, what testing do you want done, but to help define, build out, and then implement comprehensive strategies, ultimately to support regulatory filings and products. So just as a quick introduction to what we'll be talking about today, I wanted to review some very general regulatory context, both for the US markets and the EU markets. And so what I've done here is pulled some excerpts from relevant regulation pertaining to what we'll be talking about today. And so the first example here is from the 21 CFR Code of Federal Regulations here in the US. And very clearly, there are requirements, regulations that state that after filling, sealing must be performed in a manner that's going to maintain integrity of the product essentially throughout its shelf life. And that's something that's going to be a theme, as you'll see, in a lot of regulatory requirements and also best practices. This concept of this life cycle approach, considering CCI, but also all relevant attributes of that container system from development through expiry of the product that it's containing. Another FDA reference here, 1994 sterilization validation guidance. Another point that is made such that the drug product must be sterile throughout its shelf life. So it's just another touch point in another guidance document that highlights the importance of verifying sterility and therefore integrity throughout the shelf life of that product package system. EU CGMP Annex 1 also has some language around this. This is pulled from the now effective version in 2022. This was something that was in draft for many years, going back to even before Brexit. And finally, there is the published final version. And so some key excerpts here. Final containers should be closed or sealed by appropriately validated methods. Samples of products using systems other than Fusion, so we're talking about things like vials, syringes, etc., should be checked for integrity using validated methods. And an important statement here is that the frequency and therefore also the intensity of testing should be based kind of on this scientific risk-based approach, that the testing that's being performed should be relative to the knowledge and experience with that container closure system and the data that's been generated on or with that container closure system previously. And all of that goes into developing that strategy for the specific product package system. Another excerpt here, this refers to the processes associated with finishing, storing, and transporting that sterile product should not compromise the product from a sterility perspective. And so container closure integrity needs to be considered again, not just for the final drug product, but also as a result of the processes that that drug product is exposed to, both from the manufacturing perspective and also distribution, storage, and use. And this is kind of summarized in the 1999 FDA packaging guidance, such that every proposed package system needs to be shown to be suitable for its intended use. In other words, how that package system is going to be used will dictate what needs to be verified about that package system. Ultimately, you want to prove that what you're expecting or requiring that package system to do, that that package system is actually capable of doing it under anticipated conditions of use. And so this little diagram here really summarizes that, and it's pulled from the 99 guidance document, where demonstrating suitability of a package system really is comprised of demonstrating compatibility of that package system with the drug product, the safety of that package system, the protection that that package system affords to the drug product, and then the performance of that package system. So in the case of, let's say, a prefilled syringe, that performance would be actually delivering that dose, as an example here. And so when we think about comprehensive container qualification programs, there are lots of relevant tests that could be put together to overall qualify that package system. And so the purpose of this little diagram here is to try and put some, visual context behind what types of tests are relevant in the overall process of qualifying that package system. And so under the umbrella of container testing, there are both compendial tests that need to be performed or considered. These are things that you find directly in the USP, the EP, JP, BP, et cetera, other compendial bodies. And also tests that are not necessarily compendial in nature, not prescribed by the USP or the EP, but that directly relate to demonstrating the quality or the qualification, rather, of the package system. And that can be things like taking dimensional evaluations of different packaging components, different ISO and ASTM test methods. That also includes distribution simulation according to ASTM and ISTA standards. So challenging that package system to put it through the full distribution simulation circuit and demonstrate that critical quality attributes are maintained. And then also the concept of package and process development and validation, which is not something that's explicitly mentioned or prescribed in the USP or EP, for example, but ultimately a lot of the concepts that are proposed in these compendia can go into the process of development and validation. So we'll go through an example comprehensive package for a cold storage vial product, and we'll touch on these relevant tests as they become relevant through the life cycle. But we're going to take specific focus on container closure integrity testing here, because really, as you'll see from a risk perspective, CCI is at a very unique risk for these types of product package systems. So before we go too far into container closure integrity testing specifically, I want to start off with a slide that I frequently use in presentations. And it's a matter of asking the question, what is container closure integrity? When we say that a product package system has integrity, realistically, what does that mean? And so USP 1207 does provide some context. Essentially, integrity can be defined as the degree of package protection that's demanded by the product to ensure that everything physicochemical, microbiological, all the quality attributes of that product are maintained throughout the life cycle through product expiry and use. This can be fairly lengthy, so to shorten it down, we can say that CCI is really the degree of package protection that's demanded by the product. And to shorten that one more time, it's demanded by the product. So what it means to have integrity really can change based on the product, based on that package system, and based on the intended use case or the experience that that product package system will have throughout its life cycle. And so what we wind up at is this concept that it depends. to say what container closure integrity is really going to be dependent on the case that you're looking to apply it to. And so here's a good example of how the definition, so to speak, of container closure integrity might change. This is a concept that's also in USP 1207 and something that sometimes we see a little bit of confusion around, which is the concept of the maximum allowable leakage limit. So a maximum allowable leakage limit could be summarized as the greatest size leak that could be present without having a negative impact on the quality attributes of that product. So for example, if almost all package systems must maintain that dosage form. We don't want the dosage form, whether it's something really basic like a solid oral dose or something high risk like an injectable. We don't want that product, of course, physically leaking out of the package. That certainly has implications for the quality of that product and also the dose that might be present as a result of that leakage. To take it one step further, there are many products where also, in addition to maintaining the dosage form, we have to maintain sterility. And so now the leaks of concern that we're considering are any leak that's going to be capable of allowing microbiological ingress or microorganism ingress. And so that type of leak can be much smaller than a type of leak that's going to let liquid visibly pour out of that package system. Of course, here, the reason that we're worried about this for certain products is that we're worried about failure of product sterility. And the example failure mode would be that a microbe could contaminate that product if, in fact, that leak is present. To take it, one step further in terms of sensitivity, there are many product package systems where, yes, it needs to maintain sterility, but it also must maintain a critical headspace. And so this could be things that are backfilled with nitrogen because they're oxygen sensitive. This might be under pressure in the vial resulting from a lyo product, which is required to allow for reconstitution. And so maintenance of critical headspace. Now, for these types of products, we're concerned with any leak that could theoretically allow for gas exchange with that product package headspace. And again, this type of exchange can happen through pathways that are even smaller than those which microbes can transgress. So in terms of defining a maximum allowable leakage limit, CCI really is mostly discussed in the context of sterile parenterals. And so realistically, the maximum allowable leakage limit for most products that we consider is at that sterility level or below. And that's really what we'll be focusing on here, small leaks of concern. As a precursor to that, just to circle back to this life cycle concept and approach, this is something that is in numerous guidance documents, but also explicitly in USP 1207. For those who aren't familiar with USP 1207, this is the USP guidance chapter on container closure integrity, how to consider it, how to implement it. It's very conceptual in nature. It's not prescriptive. It does not by any means provide you a roadmap of what you have to do in every case, but instead provides some general concepts for consideration as you approach the integrity of a given product package system. And one of the key elements of USP 1207 is this concept of the life cycle approach, explicitly mentioning that package integrity must be considered at least really 3 product life cycle phases: the development and validation of that product package system and the processes that lead to that package system, during manufacturing, and then during shelf life and stability assessments. Again, covering that full life cycle of the product in which we consider CCI. And so when we think about that full life cycle, the concept of a risk assessment becomes very relevant. We would like to think, as we move through this life cycle of this product package system, what can go wrong at these different stages? As we go from component manufacturing to filling and sealing to shipping and storage, what are the different things that need to be considered at these different life cycle stages? How severe, probable, and detectable are these risks? And how do we go about mitigating them by a quality by design approach? And so as an example, here, this is a slide from a PDA training course that I've given in the past. All of these different things go into the risk assessment: the materials that we're using, the processes that we're using to assemble this package, we evaluate, what can go wrong and have a negative impact on the product at these different life cycle stages, and how do we implement a test regimen or a control strategy to mitigate that risk. So just as an example, and we'll certainly go into more detail in the next few slides, an example here would be for a pre-filled syringe molding defects on plungers, which could cause a CCI issue. And so one control strategy that might be implemented is 100% visual inspection of those plungers or an incoming material sampling plan to verify that those defects are not present in the lot that's going to be used for manufacturing. And so we'll go through some examples of how we approach this specifically for a cold storage product and how we implement those different test regimens or control strategies throughout the life cycle to mitigate those risks. So before we go into specific considerations for cold storage products, ultimately the example we'll be focusing on here is a vial package system. And so I think it's really important to review, how a vial creates its seal and what types of impacts different variables can have on that seal. And so when we consider leakage in general, it's well established that every package is going to leak to some extent. Leakage is a continuum, and so to say that it leaks or does not leak is one thing, but it's also at what leak rate or how intense is that leakage that we're seeing, and that goes back to the maximum allowable leakage limit. Permeation is also something that can be relevant here, whereby it may not be directly leakage. However, permeation is also going to have an impact, for example, on the headspace content of a package system. Ultimately, the drug product is not going to care whether that oxygen came from a leak pathway or from permeation if that product is oxygen sensitive. Leakage, however, is typically through one of two areas: through a physical defect in that package system. So, excuse me, leakage really can stem from two different areas. We can have leakage literally as a result of a defect through a crack, a pinhole, a tear, a defective component that's not creating that seal. But also we can have leakage through actual seal areas themselves. So what affects the quality of a seal? Leakage at a seal can be influenced by many factors, including dimensional tolerances of components, how those components physically fit together to create that seal, how those crimps are applied, how those components are mated together through the crimping process. Temperature, as we'll see, is certainly something that can impact the quality of a seal. Debris, any debris on that component that could interfere with that seal, create a channel, of course, can have a problem or an impact on quality. Innate package materials could impact this quality of a seal. And then, of course, other processing and manufacturing variables. For example, gamma irradiation can have an impact on the way that that elastomer behaves, which in turn can impact the quality of a seal. And so as we go through and we think about building a product package system that's going to be able to be suitable for its intended use, we need to consider to a large extent these attributes and how they come into play. So if there is in fact a poor seal present, how can that go about affecting the product? So of course, as we talked about in the maximum allowable leakage limit example, we can literally lose the product from the package system, which is inherently an issue. We could have a loss of sterility, a loss of potency, an increase in concentration of API as a result of solvent loss. In the case of lyo products, we can have cake collapse, deterioration, and moisture absorption. In the case of headspace or gas-sensitive products, we can have an oxidation of API if oxygen gets in that package, a pH shift of a liquid solution if CO2 enters that package and then dissolves into solution. We could have a partial loss of vacuum leading to overpressure upon reconstitution, and likely many more potential failure modes associated with a poor seal. So how do these issues arise? How do we wind up with a poor seal or a leaking package system? Well, they can be issues from the component level. This could be defective components that maybe have a physical defect that prevents them from sealing well. These components could be poorly designed. It could be that the components don't mate well together given the dimensional tolerances of those different components. Or there could also be just poor quality control of those components. There might be dimensional tolerances established, but the manufacturer might have difficulty actually ensuring that all of the units produced adhere to those tolerances. On the system level, thinking about that fully assembled package system, how do issues arise? We could have poor equipment quality or operation. We could have a lack of process validation, perhaps not necessarily looking at how those samples are crimped and yielding to poor seals. And again, component incompatibility as a result largely of dimensions of those different components. So ultimately, issues arise from some deficiency in the package or process development phase in most cases. We should note that die ingress and the twist test, simple act of taking a crimped vial and twisting that crimp seal to see if it's tight, is not suitable to determine seal integrity. These tests are not sensitive. In most cases, they're not even applied as leak tests, and are really unable to differentiate between very subtle differences like dimensional variation of components. And so realistically, these things individually, as we go through the life cycle of this package system, are not sufficient to really demonstrate the integrity throughout the life cycle. So how do we go about characterizing and ensuring a good seal? Of course, we'll need to consider the way that these components fit together. There is also consideration for the way that that stopper is compressed through the manufacturing process. Evaluation of residual seal force might be relevant, and also, of course, direct evaluation for CCI by a sensitive method. In most cases, that's helium or headspace analysis. In the example today, we're going to be talking about headspace analysis. And so the key here, and something that we'll come back to and we have already touched on, is planning. Really considering these things upfront from package development to ensure that they're not an issue in final produced product. If you're waiting till production batches to consider these things, it's really too late, to capture these risks before there's a potential impact to the product that you're manufacturing. So in terms of evaluating the integrity of a vial package system, removing the concept of cold storage for the time being, we should consider different sealing points of a vial. How does a vial actually establish a good seal? And so, of course, there's three relevant components. We have our vial, we have our stopper, and we have our crimp seal. And when we put these components together, these really yield three sealing areas. We have our valve seal here, which is where the plug of that plunger compresses against the mouth, inner diameter of the mouth of that vial. We have what's referred to as the transition, which is this point here, just at the apex as we go into the mouth of the container and we transition from the land of that plunger or stopper to the plug of that stopper. And then lastly, we have the land seal here, which is where the land of that plunger compresses against the land of the vial. And so considering each of these individually, this valve or plug seal, as it's referred to, its real function is to position the closure into the container prior to crimping. And in some cases, it can be relied on to provide integrity prior to crimping. This is something that's especially relevant for, let's say, lyo products, as it goes from that lyo chamber to the crimping line and must maintain that nitrogen backfill and under pressure inside that vial. So some key considerations, the efficacy of this seal is largely related to the tolerances of these component dimensions. And for that reason, it's typically not very robust as a seal. The land seals, however, this is where we typically achieve a primary seal for vial package systems. This is really the seal that's responsible for maintaining CCI through the shelf life of that product system. Some key considerations here is that the land seal and the quality of this land seal is really a result of the crimping process. So instead of kind of being innate to those package components and just a function of how those components fit together, here a very critical parameter that's going to lead to a good land seal is going to be the process by which that seal is applied. And because it is a process that's yielding this seal, then we know that it can be controlled, it can be measured, and to a certain extent, it can be predictable. And so there are numerous dimensions also that will impact this compression and therefore this seal as a result of that process. So we have dimensions like the stopper flange thickness, the vial flange thickness, the neck diameter, and the aluminum skirt of the crimp seal, all of which, as you'll see in a few slides, can have an impact on the compression of that stopper and therefore the seal of that package system. So to just recap briefly on these dimensional impacts, lack of consideration for dimensional extremes can lead to CCI issues. Again, this is not something that's unique to cold storage product. This is something that is applicable to really any vial package system. These issues can exist even if a validated deterministic method is used for routine testing of final production samples. So again, just verifying that a certain number of production samples at the end of the day aren't leaking does not necessarily negate the need or the impact to evaluate these concepts in package development. And ideally, these dimensions, these component dimensions, should be built into capping optimization and process validation. So, excuse me, when we consider this specific to cold storage products, we have all of these same similar considerations to other sterile injectable vial systems. All of the things that we've talked about to date, or to this point, are still relevant here. However, we have additional considerations due to the impact of cold storage. So again, what we're looking to do is demonstrate this package system as suitable for its intended use. And a very critical aspect of this package system's intended use will be to maintain integrity during distribution and storage at ultra cold or deep cold temperatures. And so, what is the problem? What is the additional consideration that needs to be evaluated specific to cold storage package systems? And really, it has to do, as we've touched on before, with that temperature. So at or below approximately minus 60 Celsius, elastomers that are used in a lot of traditional package systems can reach glass transition. And what this can lead to is elastomer shrinkage and thus leakage while these containers are stored at that ultra cold or deep cold storage condition that does not necessarily present itself when we're at room temperature. And so I have some photos here from some work that was done by Chris Folta at Janssen back in 2016 and presented that highlights this. And so this top image here is a depiction of what happens to this stopper when we're at room temperature, which is the outline, and then once we're at or below the glass transition state of that elastomer. And so you can see physically that this elastomer has shrunk. And so when that's part of a compressed package system, the same thing happens. And this second photo here shows exactly that, but the stopper applied and crimped down to a vial that is being brought down to ultra cold temperatures or deep cold temperatures. And again, you see that the stopper has shrunk a little bit relative to the vial and the crimp seal. And so what this can lead to is leakage through small pathways that are present while we're at ultra cold conditions, but that when we take them back up to room temperature, which is where most traditional CCI measurements are taken at room temperature, that elastomer has the chance to warm up, reflow, regain its elastomeric viscoelastic properties, and then reseal. And so what this yields is what we refer to as transient leakage. leakage that occurs, but it's not a permanent leak. It's not a physical defect in that package system like a crack, a pinhole, or some fibers that cause a channel, some debris that causes a channel. This is a transient leak. It can come and go based on the conditions that the package system finds itself in. In this case, of course, that's temperature. And so if we revisit our comprehensive CCI risk assessment, we ask ourselves, what can go wrong at these different phases, and how do we go about controlling or mitigating those risks? And so what I've done here, just very, very high level, is outline kind of typical considerations for vial package systems at these phases, and then what additional considerations we take for cold storage package systems. So from the component manufacturing perspective, of course, we're always concerned about component defects, deformed stoppers, etc., and also dimensional tolerances. But for cold storage, these elements have an even greater impact or risk to CCI because of what can happen at those ultra cold temperatures. During filling and sealing, typical considerations for vials, of course, include the machinability and component stackup, how we go about crimping those vials based on the compression of that stopper and its residual seal force. For cold storage applications, we need even tighter control in this area because even packages that are integral at room temperature may leak once they're placed into cold storage. And so there's extra scrutiny placed on these attributes, this process and these package components for cold storage applications specifically. In shipping, storage, and use, typical considerations for vial package systems include the impact of physical shock and stress as that box, let's say, or shipper of product is, shipped around, vibrates on trucks, airplanes, etc. We similarly consider altitude and pressure and temperature. Component aging, just as a function of those components being used and then on a shelf, let's say, for 3 years. Any potential product package interactions. But of course, for cold storage products, key consideration is that transient leakage. That transient leakage due to component shrinkage at deep and ultra cold storage temperatures. And so it becomes extremely relevant to consider specifically the storage temperature as we move through these different life cycle stages. And so how do we go about actually doing that? How do we go about implementing, devising a program for cold storage package systems? This here is an idea of the types of technologies and instruments that we would use here at CS Analytical to perform this type of program. So we will use our Keonce dimensional analysis system, which is pretty much a video microscope and an optical comparator in one. So some system that's able to evaluate, measure the dimensions of vials, stoppers, and crimp seals. We have typically lab scale capping, but industrial style. So we maintain a Genesis Integra unit, which we can outfit with change parts and different parameters to crimp in a laboratory setting vials under controlled conditions. We maintain residual seal force testers, both Genesis and Instron, a full suite of distribution simulation equipment by Lansmont for vibration, drop, and compression, an altitude chamber, minus ADC, dry ice, and liquid nitrogen storage capabilities are required. And then in this case, we're using Lighthouse CO2 and oxygen headspace analysis systems. So just an idea of the type of equipment that we use here in-house to perform these types of programs. Of course, similar types of equipment can be used to achieve the same study goals. So let's start moving through the actual life cycle of this product package system. And what we'll do is at each of these different life cycle stages, we'll talk about what's relevant, what type of testing or control strategies can be implemented, and also why we're doing that, why we're recommending that, and what types of risks what we're proposing here helps mitigate. So in this package development space, looking at things on the component level, there's a lot that can be done already, just looking at individual components. And our goal here is to really select suitable components that are going to yield an integral package that's going to be suitable for its intended use from all perspectives, including leakage, and really establish a final component and package design space. So some basic compendial tests that are relevant here in this package development phase: USP 660 on glass, USP 381 and 382 on the elastomeric components. If a plastic vial is being used, then also USP 661 would be relevant in that case. All of these tests are intended to look at the physicochemical properties of that component, with the exception of 382, which in 2025 will be broken out from 381 and focuses specifically on the functional performance of that elastomer. And so we typically recommend that the basic compendial USP EP tests are done in the package development space so that you know that ultimately if that component is chosen, used moving forward, that it is capable of meeting all of the relevant requirements in that compendial test chapter, and there's no issue, at least from that perspective, for regulatory approval. Also in this package development space on a component level, we want to look at dimensional analysis and stack-up tolerances. So we've touched on this before. Now we can talk about how we actually go about doing that and why it's important. And so what we're talking about doing here is evaluating dimensional variation of components within a lot, let's say, of stoppers, a lot of vials, and how these dimensional tolerances stack up. When we place these different dimensional extremes together, how do they impact the integrity of the package system that's made from those components? And really, what evaluating this does is allow us to integrate dimensional impacts and dimensional variation into our capping studies for process development and process validation, but it also allows us to establish or verify the manufacturer tolerances for these components. You know, when there are stoppers being made, it's not always the case that a certain 20 millimeter stopper is going to work very well with a certain 20 milliliter vial. It's not always that the two are designed in tandem. And so evaluating the dimensional tolerances can help choose your components, but also can help nail down what is an acceptable dimensional tolerance for routine manufacturing lots moving forward. And so we talked about how these dimensions are relevant previously for the seal. Here's what we're actually looking at in this design phase and development phase. And so for the valve seal, when we have the widest vial mouth inner diameter and the smallest plug diameter that are within the dimensional tolerances as established by the manufacturer, do these dimensional extremes yield a good seal? Is there enough overlap to create what we call an interference fit, such that the elastomer would be compressed against the opening of that vial mouth? On the other side, the other extreme, let's consider the smallest vial inner diameter of the mouth with the widest stopper plug outer diameter. And so when we have these other dimensional extremes, does this stopper even fit into the vial? If it does it pop back up out of that vial as a result of too much compression in this interference zone? Or do we start seeing deformation of that elastomer, let's say wrinkles or puckering, as a result of this aggressive dimensional overlap? And so by taking measurements on a wide number of stoppers and a wide number of vials, we can put those components together to specifically evaluate the risk to integrity at the dimensional extremes that these components may come in. Considering that land seal, similar concept, we're looking at minimums and maximums, and we're looking at the combinations of these minimums and maximums and their impact on integrity. So as an example, if we take the minimum stopper flange thickness, the thinnest stopper flange within dimensional tolerance range, and the minimum vial flange thickness, smallest distance from here to here, and we use that with the maximum skirt length, so the maximum allowable within tolerance aluminum crimp seal skirt length. Does this package system seal? And if it seals well, does it seal well? when we think about this minimum stack up, given a constant set of crimping parameters, we might yield low compression in this case as a result of these minimums. We might also see that when we go and apply that crimp and tuck this skirt under, because this skirt is so long and this vial and stopper flange are so short, there's a lot of excess material hanging out. And so when we go and actually apply this crimp seal, do we get contact with the neck of the vial, which can, one, introduce things like scoring or cracks, or two, prevent that crimp seal from being properly applied and adequately compressing that stopper against the glass. Looking at the maximum stack ups, again, if we have this thickest stopper flange, the thickest vial flange, and the shortest skirt, are we able to compress that stopper against the glass enough that that skirt can tuck under and maintain that seal? Or do we get compression that is just too high that can lead to cracks and other issues in the vial as a result of these dimensional extremes? And so when we think about that at a component level, now how do we go about evaluating that on a system level, that product package system? And so here, what we're trying to do is define and optimize the design space of that package system and demonstrate suitable performance under actual conditions of use. And so here's where we're putting together vial package systems, possibly at different crimping parameters, and evaluating, how does this vial package system seal when we apply certain types of parameters, and ideally when we're incorporating dimensional extremes. And so oftentimes we'll do this lab scale using automated hand or an industrial style capper, such as the Genesis Integra, allowing us to crimp at a range of forces. Ideally, we will have samples prepared that also reflect the dimensional extremes at each of these crimp forces. So we get the full range of dimensional variation within each sealing set, but we're looking at how those different package systems perform from an integrity perspective after they've been crimped according to different parameters. And so doing this, we can actually calculate in the laboratory percent compression of that elastomer based on the dimensional tolerances, stack ups rather, that we measure before and after sealing. Residual seal force testing is often incorporated into this package development phase and provides a way to correlate leakage and compression back to RSF. For those who aren't familiar, residual seal force is essentially an indirect measurement of how much force this stopper is applying to the top of that vial. And that's going to be a function of how compressed that stopper is, and ultimately that can be correlated to whether or not that vial has an integral seal. The last part of a capping optimization study would be, of course, to implement and integrate, rather, container closure integrity testing. And so in this case here, we're talking about headspace analysis using either CO2 ingress on dry ice, oxygen ingress during liquid nitrogen or liquid nitrogen vapor phase storage, and then lighthouse headspace analyzers to measure change in headspace concentration over time. Just to give some background on the principle of operation here for headspace analysis, let's just consider an air-filled sample with about 21% oxygen and very low CO2. If I place this sample into a dry ice environment, as would be the case for, let's say, products stored at -60 or below, during shipment, typically that incorporates dry ice. So when placed in dry ice conditions, the environment is CO2. As that dry ice sublimates, it's going to create a CO2 environment. Any leakage that's present in that vial is going to allow exchange of that headspace with the CO2 environment. And so we will lose air headspace and have that replaced with CO2 as the dry ice sublimates and that gas leaks into the package during storage. And so we can measure this increase in CO2 after storage on dry ice as an indicator of whether or not that sample leaked at these conditions. Similar setup for samples placed in cryogenic conditions. except in this case, the environment is nitrogen. And so in this case, we're replacing the air headspace with nitrogen, and a decrease in oxygen is what we're measuring. And of course, a decrease in oxygen from about 21% downward as a result of nitrogen replacing that headspace is indicative of leakage. And so the key advantage of implementing headspace analysis in this case is that we're able to detect what we call those transient leaks. Even after we remove that vial from the liquid nitrogen storage environment or the dry ice storage environment, bring it back up to room temperature, even if that elastomer reflows, reseals, the headspace is trapped inside of that vial. And we're able to measure and therefore detect whether that vial did leak did leak at cold storage conditions, even if it's not presently leaking at room temperature. Moving into process and package validation, here our main goal is to demonstrate the suitability of the system under intended production and distribution processes. So we've characterized it kind of in that laboratory environment during that package development phase to really define our design space, to define the machinability of that vial package system. And here's where we're actually transferring that knowledge, leveraging that knowledge to demonstrate that the way we're producing this package system, whether it's in clinical or commercial manufacturing, is going to yield those integral containers the same way that we saw in that development phase. And so this is where this concept of capping line qualification becomes very relevant. So leveraging that data from the development phase, ideally we can use the percent compression evaluation and residual seal force data correlated to integrity to scale and transfer those capping operations. So if we demonstrate in development that a certain set of capping parameters leads to very integral package systems, the intention here would be to replicate those capping parameters on the actual clinical or commercial production equipment. And so to actually qualify this line, having that data from package development makes setting up these parameters that much more straightforward, and also informs or makes it even more straightforward to verify that integrity is maintained by running a similar type of measurement on actual units that were produced on that production equipment, evaluating the residual seal force of those samples coming off that line and challenging them by a CCI headspace analysis method to determine whether or not those samples under actual production processes are also yielding integrity. At this phase, we typically consider full development and validation for headspace analysis methods. This is because once we have that validated method, it can be used in a plethora of ways. So we can use this to support routine testing of packaging from the line, incorporate this method into studies that we'll touch on in a moment here, but also as a routine QC or stability test if and where appropriate or necessary. So once we have that validated method, one key example of how we incorporate that valid method into other studies would be distribution simulation. And so we talked previously about the distribution process, how shock, vibration, etc. could have a negative impact on product package systems, but also we know that shipping that around the world in a dry ice container or in a liquid nitrogen dewar adds a whole nother layer of complexity and risk to the process. And so what we are able to do is once we have that developed and validated method, and ideally have some data that show that the package system should be integral the way that we're assembling it, now we can go about actually packing out secondary and tertiary package systems with this primary package system, subject them to distribution simulation, drop, vibration, altitude, etc., and follow that up with container closure integrity inspection, testing, and visual inspection. And so what we're able to do is take this validated method, demonstrate that all package systems have integrity before distribution, subject them to the challenges of the distribution simulation, and then follow up with a confirmatory container closure integrity evaluation to prove that the distribution process is not going to have a negative impact on the quality of that package system. Also in this phase, we're considering some other less routine and more custom USP or compendial tests. And this would be things like extractables and leachables, and also USP 1660 glass delamination evaluation, if in fact you are using a glass vial. So as we move into actual manufacturing, whether that's clinical or commercial, our goal in this life cycle stage is to demonstrate continued control over the variables that can impact seal integrity, right? So we have this body of data that's showing the package system and the components, when we put them together, yield integrity. We should have data that show the way that we're manufacturing these samples, or excuse me, these package systems yield integrity. And here what we're doing is implementing control strategies to ensure and monitor that that is the case throughout the manufacturing process. So some examples of that would be incoming QC inspection of package components. That would be both visual inspection for deformities, let's say, but also that dimensional evaluation, harkening back to the package development phase. Verifying on a lot basis that the materials we're receiving fall within the dimensional tolerances that we verified during the package development phase. Again, minimizing risk by implementing a control strategy that directly addresses variables that can impact seal integrity. Oftentimes, we see USP 660 and 381 implemented on a lot-by-lot basis or part of the incoming QC inspection for those glass vials and those elastomeric stoppers independently. There's also this concept of in-process testing. So one thing that residual seal force is frequently used for is monitoring sealing consistency off those capping lines. Excuse me. So if we have this work that was done in the development and process validation space, where we can clearly show that samples within a certain residual seal force range correlate to integrity, then we can use residual seal force as kind of an in-process check to ensure that our different capping lines are yielding similar residual seal force values, but also over time, we're not seeing a lot of drift with respect to how those samples are being capped. Just as a note, residual seal force is not an integrity test, but absolutely can and probably should be applied as kind of that control or quality assurance practice. Lastly, for in-process testing, some companies choose to implement a sampling plan by headspace analysis for samples actually coming off that line. The actual application of this particular sampling plan is always going to depend on the product, the package, a lot of different considerations, but having an in-process CCI test is not necessarily outside the realm of possibility. Again, considering, let's say, EU CGMP Annex 1, the extent that you go to when you think about routine testing for CCI really should be based on all of that historical knowledge. And at this point in the process, hopefully, as you've seen through this presentation, we should have a really robust data set of historical knowledge on this package system and these components that helps frame and minimize the amount of routine testing we have to do. Moving into our shelf life stability assessments, our goal here is to demonstrate the maintenance of integrity through expiry and use. And so this is where our validated CCI method, in this case headspace analysis, is typically performed at real-time and accelerated aging time points as part of a stability protocol. And this is done with USP 1207 best practices in mind, but it's also an approach that's explicitly mentioned and even recommended in a 2008 FDA guidance, CCI in lieu of sterility after time 0. So if we reconsider this diagram here, outlining comprehensive container qualification programs, you can see that there is a lot that needs to be considered, but when and where these different tests are considered really is going to change based on the life cycle stage. And considering all of these attributes, all of these tests to demonstrate qualification only at the end, once you've manufactured actual production batches, is largely infeasible because you can't necessarily go back with ease and, let's say, change that stopper, or go back with ease and reevaluate or redefine acceptable dimensional tolerances for that stopper. And so what we've done is throughout the life cycle, we've mitigated those risks using that quality by design approach applied to that product package system through its life cycle. Again, considering the components, how those components fit together, and all of the variables that can impact that package system once it has been assembled. To switch back to our little diagram here, specifically for cold storage products, you can see as we move through these different life cycle stages, how we go about actually painting that overall picture of package qualification. So some quick conclusions. Packages stored in deep or ultra cold storage temperatures have additional considerations or additional risks compared to traditional vial package systems stored at normal temperatures, room temperature, refrigerated, or even -20 frozen. The risk of transient leakage puts increased importance on component dimensions and sealing processes, all of which are evaluated at room temperature in most cases, but have different impact when we put them at cold storage, and those dimensions can change. And so it's increasingly important relative to vials stored at other temperatures to consider these dimensions, to characterize these dimensions, and the process by which these package systems are assembled. As we discussed, evaluation of leakage at room temperature does not impart integrity at cold temperatures. This is something that's really critical. I have seen numerous cases over the past few years where we have clients who are storing at deep cold, ultra cold storage temperatures. They've approached a contract lab saying that they need a CCI method, and they wind up with a vacuum decay test. Now, vacuum decay is a fantastic tool. It's part of our overall CCI toolbox that we use. However, in this specific instance, vacuum decay is looking for leakage while that sample is at room temperature. And so testing that sample at room temperature and demonstrating it has no leak does absolutely nothing to say whether that package system is going to leak once it's at these aggressive conditions. And so directly evaluating leakage as a result of that storage temperature must be incorporated as part of the overall qualification process. And the best way to do that is with that quality by design approach, characterizing, mitigating those risks through proper development, validation, and control strategies. Just a few outlines of some program advantages for how we implement these types of programs here at CS Analytical. All of these services that we've discussed today are completed through a single service provider in a one-stop shop model. So we have the ability to take it from that package development component level evaluation up through and including stability analysis. All of our services are performed in an FDA registered cGMP facility, can be incorporated into regulatory filings as needed. Of course, we have the unique ability to leverage CS Analytical's internal expertise, but also our industry relationships. Within the niche of container testing and CCI specifically, CS Analytical really has some people who, outside of some folks who are really leading experts on this type of concept, and have that tight-knit relationship with other people in similar roles throughout industry. And so we're able to leverage, our internal expertise, but also those broader industry relationships to provide a successful program. And lastly, the approach to this is flexible. So it's not always the case that we have a client come and contact us in that package development phase. They may already be in clinical phases, or they may already be preparing for commercial. And so this program can be designed and scaled to include services that really meet the needs of the client in that moment, or sync with the life cycle stage that product's in, and sort of reevaluate the risk within the context of that specific product's situation. So with that, I appreciate your time and attention, and would be happy to take any questions that our attendees may have. If you would like to ask a question, just feel free to raise your hand using the meeting, or just send something in the group chat for this
Yeah, there
meeting.
was an earlier question in chat, Brandon, from Steven.
Yeah, Brandon, it's Brandon, Steve Oros. How are you?
Hi, Steven. Doing well, thanks.
Yeah. In your evaluation of things that can cause land seal concerns, mismatches in just the dimensional mismatches, I've also seen surprisingly twice just this year a lack of evaluation of fill volume in instances where you're freezing the product. For example, filling the 10 ml vial to 10 mLs and then putting it to -60 and then having obviously CCI or seal issues. Just wondering if you've seen that before, and that might be something you'd consider as part of the process evaluation.
That is a good point. So yes, of course, when we have a liquid product that's put under frozen conditions, that product will have a tendency to expand. And so we have seen circumstances where that fill level, if it's too high, can expand and interfere with package integrity. Actually, that's something that we see more commonly in things like IV bags or cryo bags, bioprocessing bags. If they are overfilled, put into these frozen conditions, which can include deep cold and ultra cold storage conditions, it's almost a presentation in and of itself. But where that product expansion can lead to cracks or tears in that package system, which of course compromises the integrity. So certainly a good point, well received. And yes, we have seen areas where high fill level specifically can yield integrity issues down the road.
Brandon, maybe just a question. Have you seen in particular any issues with the polymeric components, especially with the deep cold storages?
Sure. Yes. Long story short, I mean, when you're referring to polymeric components, are you referring to things like vials, COC vials specifically?
Yes.
So the same concepts are relevant for polymeric primary package systems, because really the bulk of what we're seeing in terms of issues at these cold storage temperatures really comes from that elastomer and not so much from the glass, let's say, side of things. Now, one thing that's worth considering is that in some cases, polymeric components also will have a tendency to shrink at ultra cold storage conditions, perhaps more than glass would. So there is some interplay for, let's say, a COC vial, where that shrinkage of that vial, even if it's ever so slight, can help offset the shrinkage of that stopper, so to speak, if you compare it to glass, which is staying the same as that stopper shrinks. But generally speaking, we see these same types of considerations, concerns, and potentially issues, regardless of whether that vial is polymeric or glass.
Okay. And in terms of permeation, do you see also any different behavior?
Yeah, so I have not done a lot of hands-on work personally looking at permeation differences between, let's say, a 2R polymeric and a 2R glass using the same exact elastomer. However, generally speaking, I would expect that polymeric components are going to be more susceptible to permeation than glass components would, given a constant elastomer. However, there are also a lot of polymeric vials that have coatings on the inside that can really limit that. So, for example, SiO2 coatings would help, offset some of that permeation that could theoretically be relevant in a polymeric package system.
Okay, thank you.
Any other questions from the group here? All right. Well, if nobody else has any other questions, then I guess we can more or less end here. And by all means, if you do have any questions, please feel free to follow up. There's our email address that goes to our general inquiry mailbox. You can feel free to reach out to us at any time to talk about any of the concepts we discussed today, how these concepts might be applied to different types of package systems or storage conditions, or a specific case that you guys are working with currently and would just like some guidance on. As I mentioned earlier, we really strive to be collaborative in that approach and help people define their strategies before they implement them. So by all means, feel free to reach out to anybody at the team here at CS Analytical. And with that, again, thanks everybody for joining. I appreciate your time and attention and hope to speak soon.